Hello, this is Amanda Welch welcoming you to this Bite-Size Bio web seminar, which today is sponsored by Bio-Techne. Today's presentation is titled Isolation and Culture of Primary Cells from Bone Marrow and is being presented by Dr. Peter May from the University of Connecticut Health Center. Dr. May received his PhD from Wesleyan University in 2001, where he studied the role of hedgehog signaling during embryonic stem cell differentiation and early mouse development. He then carried out two postdoctoral fellowships at the University of Connecticut Health. The first postdoctoral study focused on mechanisms of hedgehog and Wnt signaling. The second postdoctoral study focused on skeletal biology with particular focus on osteoprogenitor cells. Currently, Dr. May is an assistant professor in the Department of Reconstructive Sciences, and recently he was appointed as Associate Director of the Skeletal Biology and Regeneration Program at UConn Health. His lab often uses developmental genetic approaches to study skeletal biology. Active areas of research in his lab include stromal cell biology and the differentiation of pluripotent stem cells into skeletal progenitors. Now, as always, we will have a question and answer section at the end of the presentation. So please type any questions that you have into the questions box, which appears on the right-hand side of your screen, and I'll put them to Peter at the end. So now over to you, Peter, for the presentation.
Thank you very much, Amanda. Welcome, everybody. The title of my talk today is Isolation and Culture of Primary Cells from the Bone Marrow, with an obvious focus, as sort of Amanda alluded to stromal cells of the bone marrow. This is sort of a mixed bag of sort of bringing out sort of technical issues with culturing primary bone marrow stromal cells, as well as I'll highlight some really nice studies in the literature that not only that other people have done in addition to my lab, that really, I think, have shown the progress of our understanding of what stromal cells are as well. So let's start with some very basic concepts of stromal cells. I think it's really important to understand where the cells come from. They obviously come from the bone marrow, and they're a component of the bone marrow that's called the stroma. And the stroma is actually everything that's just non-hematopoietic, okay, in terms of cell lineages. We know the bone marrow has a source, two stem cell sources, the hematopoietic stem cell, and what's typically called the mesenchymal stem cell, although I think people have kind of skirted away from that term. And I'll go through the sort of the chaotic nomenclature of stroma cells in my next slide. But the stroma consists of a vast variety of tissues, osteoblasts that line the bone surfaces, endothelial and smooth muscle cells. The endothelial is actually pretty interesting because the bone marrow has basically sinusoids. So these are fenestrated endothelial pockets where blood cells move in and out of the bone marrow. And actually, stromal cells are frequently, and it's almost like a characteristic of stromal cells to line these sinusoidal blood vessels. You obviously have the innervation of the bone marrow as well, and then you have adipocytes in addition to osteoblasts. And it's generally thought that the stromal cells give rise probably to both the adipocytes and the osteoblasts, and therefore provides a resident progenitor. And of course, the stroma provides a very important, stromal cells provide a very important niche for hematopoietic stem cells, and also orchestrating certain elements of hematopoietic differentiation. So, I thought I would just, go over nomenclature, because I would say this is probably, in terms of the field, it's incredibly problematic. Nomenclature remains one of the worst elements of understanding stromal cells or mesenchymal stem cells. And I think part of this is trying to or at least I think it's a symptom or a sign of how much confused the field is about this cell population, and how also our thinking is evolving over time. Certainly mesenchymal stem cells is probably the most common term often used for stromal cells. I think nowadays maybe more mesenchymal stromal cells is used, but connective tissue progenitors, skeletal stem cells, just plain old stromal stem cells, stromal progenitor cells, mesenchymal progenitor cells, and even CAR cells have been used to sort of refer to stromal cells or maybe a subpopulation of stromal cells. So the literature can be incredibly confusing, and I'll even confess myself that in my own papers, I've gone from using mesenchymal stem cells to mesenchymal progenitor cells. And I think that involves my own thinking about what this cell population is as well. So there are some inherent biological properties to stromal cells that sort of define this population. And that is basically that they at least have tripotentiality, okay? And that they can differentiate further into the Osteoblast lineage, the chondrocyte lineage, and the Adipocyte lineages. People have talked about them differentiating down other lineages as well. They obviously have also inherent properties to support hematopoiesis, like I mentioned. They're also they reside in the bone marrow, and skeletal biologists like myself tend to think that stromal cells are really the resident precursors to osteoblasts in the bone marrow, and when you potentially damage bone, also that they contribute to the bone healing process, and there are plenty of studies that provide evidence for that. Although certainly in cases of fracture repair, the bone also has an outer periosteum that contributes heavily to the repair of bone as well. So stromal cells also display anti-inflammatory properties, and this is something I'm honestly not too familiar with, but certainly therapeutically, it's a property that is being highly sought after, and in many ways it makes sense because of its role in hematopoiesis. And I know certainly, I think there are clinical trials trying to mix stromal cells during hematopoietic stem cell transplants to prevent graft versus host problems. One of the sort of classic properties of stromal cells is their adherence to tissue culture plastic. And we'll talk about this a little bit later on, and how this is certainly true for probably a lot of stromal cells, maybe not all of them. So the other property of stromal cells is if you plate them down as single cells, that they will grow and form colonies. And so the CFU assay is a very common assay that researchers use to quantify the number of sort of progenitor cells / stem cells that exist in a bone marrow. So finally, the cell surface phenotype of a stromal cell, there's a lot of very good markers, but none of them necessarily exclusively identify a stromal population. And we'll discuss a little bit about cell surface markers as well. So one of the things that I want to talk about, just as a quick comparative, is sort of the where stromal cells are compared to embryonic stem cells. So my lab also uses embryonic stem cells in research, and there really are stark differences, not only just in terms of their potency, because obviously we know, being an adult stem cell population, that it's understood that they're going to have a limited potency versus an embryonic stem cell that's pluripotent. But there's also, I think, some very important things that we don't know about stromal cells. So for example, what are the factors that maintain stemness in stromal cells if they are stem cells? So for example, in the embryonic stem cell field, we now have both in mouse and human a very good understanding of the transcription factors that really are important for driving and maintaining the pluripotent state of embryonic stem cells. And we really, in the mesenchymal stem cell or stromal cell field, have no idea of what transcription factors will maintain the stemness or the multipotency of this population. I think one attractive possibility is that certain transcription factors that drive sort of a mesenchymal phenotype like twist or snail could be those factors. But I think really definitive evidence of those transcription factors and what controls their expression in stromal cells has really remained to be proven. So also, in having an ability to define the stemness state, what is the media then that can be used to maintain that state? And so in embryonic stem cells, there are defined media formulations. And interestingly, these conditions are slightly different for mouse and human embryonic stem cells. But there's also issues like karyotype stability as well, so the genetic stability. Whereas in stromal cell, adult stromal cells, media formulations remain largely undefined. There are some commercial sources out there that claim to maintain stemness. And a lot of this is based on doing flow for cell surface markers. I don't know of any real rigorous testing of saying we've passed these under our media conditions for 10 passages and comparing one passage to the next passage in terms of their pluripotency and genetic stability. So anyways, I think there is still a lot to be learned in terms of that. For embryonic stem cells, there is, in principle, an endless ability to passage, although that may not necessarily be true, but certainly a considerable ability to passage. Whereas in mouse and human stromal cells, there's a limited number of passages. And I don't think the number of passages is really known, and I think it's been thrown out quite a bit in terms of there's a difference. What I would like to say is there's a difference between being able to grow the cells versus maintain their multipotent properties. And I think that's something that needs to be weighed out. But there's also a few studies that talk about how after maybe 3 passages, the genetic stability of the cells starts to actually change. The actual gene expression properties of stromal cells begin to change markedly as well. So one of the things I'll touch upon, but I'm not going to get into this very in depth, is the developmental origins of stromal cells. And so this is something that my work has touched upon in the past, using inducible and constitutive Osterix Cree, fate mapping studies in the mouse. But a number of labs have done these kinds of studies. And what you are looking at here is actually fate mapping using Osterix Cree crossed with a Cree reporter mouse, AI9. And you could see that not only, so let me tell you what Osterix is. Osterix is a transcription factor, very important for osteoblast differentiation. If you lose Osterix, you don't make osteoblasts. But one of the interesting outcomes is if you actually drive Cree recombinase with regulatory elements from the Osterix gene, you not only label osteoblasts, but you actually end up labeling tons of stromal cells in the bone marrow. And so on the left part of this slide here is the constitutive Osterix Cree, which labels stromal cells throughout the bone marrow. And on the right side is an inducible study we did where we induced Cree recombinase activity at embryonic day 14.5. And now you're looking at a 2-month-old bone. And essentially you can see part of the marrow is labeled with stromal cells. And the reason why the rest of the marrow isn't labeled by stromal cells is because it's a transient induction the tamoxifen wears off, and then the bone continues to grow and develop. So anyways, but there's been a number of groups that have essentially used Osterix Cree, used other drivers such as PRX and leptin receptor Cree, and I would encourage you to read all these papers. They're excellent papers, and have really provided a wealth of information on sort of the mesenchymal origin of bone marrow stromal cells. And from a sort of a very simplistic standpoint, I find it very interesting that Osterix, a transcription factor that is very important for osteogenesis, is actually also essentially using the Cree strategy, labeling cells that contribute to the stromal cells. And it really suggests that the lineage that connects these two is very close. And so it makes sense that maybe the stromal population is a residing progenitor population for the osteoblast population. Okay, so when you flush the bone marrow from an Osterix Cree labeled mouse, you'll basically see lots of positive cells noted by the red fluorescence in panel A here. And this is taken at day 1 of culture. So you could see essentially you flush the bone marrow. Obviously the bone marrow is chock full of hematopoietic cells, and then you start to see some attaching cells that are fluorescing red. And by day 6 you'll see lots of red cells, but you'll also notice essentially that there are a lot of negative cells that aren't fluorescing red. And we'll talk about what those cell populations are as well. Some of the notorious stromal cell genes that are sort of enriched in stromal cells are CXCL12, stem cell factor, and angiopoietin-1. And this is just a sorted population of stromal cells showing that enrichment from the Osterix Cree mouse. So one of the things that I just wanted to point out briefly was typically most labs grow cells as an inherent culture. It's really one of the classic ideas behind growing stromal cells is that they're an inherent population. But there is some very interesting work from Paul Fournette's lab where he grows stromal cells as mesenspheres, so basically in non-adherent dishes, and has a great capacity to grow these cells. And I confess I have not grown them like this. And so this might be a very good way to grow them. And we'll talk about possibly why, because I have a belief that stromal cells like to stay in contact with each other in order to promote their growth. And I'll show you why later, or some evidence of why later, from a different perspective. But with mesenspheres, certainly the cells can stay attached to each other in cell clusters. So this could be a very alternative and yet very interesting way to culture the cells. So this is sort of what I thought I would just initially, before we get into sort of the technical end of culturing stromal cells in terms of going through them very methodically and stepwise, how I would go about doing an in vitro differentiation characterization to demonstrate multipotency. And so on the top panel here, you actually have our stromal cells that were differentiated under osteogenic conditions. And typically what you do is you have a comparative panel of whole mount staining for bone mineral. Excuse me. The panel below is essentially grown under non-osteogenic conditions. Excuse me while I get a little water here. Hold on. All right, thank you. And of course, then looking at markers of gene expression for osteoblast differentiation. Now, the thing that I want to point out here when we look at gene marker expression for osteoblast differentiation is the fold difference, because this is one of my sort of beefs with labs that go through and passage multiple times and then they go to differentiate cells, is they will look at genes like osteocalcin, which here is in the mouse is called BGLAP1, or bone silo protein, dentin matrix protein, and Osterix. And when you look at the fold regulation of a lot of studies, you typically will maybe see 3 to 5 fold increase, not a very high increase. Whereas in our studies, when we actually sort and replate these cells and differentiate them under osteogenic conditions, we get, several orders of magnitude increased in gene expression. So the 0 here is day 0 of differentiation, positive sorted population. So this would be in this case, the Osterix labeled stromal population versus the Osterix negative sorted population. And then we differentiate them and then detect their gene expression. And you can see here, the scale here is times 100. So 9 days after differentiation, you were basically, 2,000, 20,000 fold increase in gene expression. It's just, it's a very large increase. So, and that's not only true for osteoblast differentiation. We can look at chondrocyte differentiation as well. And the fold increase of certain genes like aggrecan and COL10 and COL2, understandably, should be very high because these are extracellular matrix, very abundant chondrocyte markers, whereas SOX9, obviously a transcription factor, shouldn't be as high, but still we're getting considerable upregulation as well. And we'll do Alcium blue staining to demonstrate condensations forming too. And then, of course, stromal cells have a great capacity to form bone marrow fat, and they readily differentiate into adipocytes, and some gene markers of adipogenesis are adiponectin, perilipin, FABP4, and adipsin. And again, the fold upregulation of these gene markers is considerable. So that's just something to be mindful of. Notice also the abundance of adipocytes that are forming in our culture system. A lot of the time, when you look at papers, they typically will show 1 or 2 adipocytes counterstained with oil red O, and demonstrating the ability of the stromal cells to differentiate into adipocytes. And I think collectively, if you're one of these people reviewing the paper, looking at the paper, you have to take into account these kinds of pieces of data. I'm just trying to emphasize rigor in reviewing work in the field. It needs to be fairly rigorous. So another common thing that really needs to be done is transplant stromal cells. And in fact, there are certainly people who culture human stromal cells that really say the only true way to really demonstrate the multipotency of human stromal cells is by transplantation. We cannot rely on in vitro differentiation. Many people certainly say that for mouse stromal cells as well. And so what you're looking at in this figure here is essentially a femoral defect that was surgically engineered. You can see that big gap. And then we took day 5 stromal cells that were labeled with the Osterix Cree reporter approach, and we loaded a scaffold, and we essentially then implanted that scaffold into that femur, and then over time, essentially, you could see that bone healing quite beautifully. And when you look actually at the tissue section of this femur, you'll see everything in red fluorescing here is a contribution from the stromal cells. And you could literally see almost the boundary of where the implant was and the new bone is formed versus where the host bone was formed. If you look at higher magnification, you will see essentially contributions that the stromal cells gave rise to, not only to bone tissue, but also to components of the bone marrow. You see adipocytes forming, essentially, that are basically labeled with our Cree reporter strategy, and also new sinusoids, essentially, in the marrow compartment as well. And this is very typical of what you would expect to see from stromal cells when transplanted. And so let's get into now the sort of isolation of stromal cells, because there are potentially different ways to do this. And we've used both of these two types of approaches in the lab. I refer to one approach as sort of the needle and syringe method, which is more the classical approach, where you actually go ahead and you, after isolating your, typically we isolate the hind limbs, take out the tibia and femur, and then we cut the ends of the bone off, and then we'll take and we'll flush out the marrow with just a needle and syringe, where the syringe is full of media, and that gets flushed into a tube. And then we will strain that after we get it flushed through typically a 70 micron strainer. The 40 micron strainer is probably even better. One of the reasons why you strain it is because this process is pretty tough. As you can see, the needle will go in and it will damage the trabecular bone, and often you can get some bone chips. So the straining can remove the bone chips from the culture. Every once in a while, you'll still get a bone chip in the culture. Another method that we have used more frequently now, and it's because in many ways it's easier and it works just as well, if not better, is sort of the centrifuge method, where again we take our femur and isolate it, but instead of cutting it on the ends, we'll actually cut the bone in the middle, and then we'll invert the bone into a holder of something. Now, one of the things that we do a lot of DNA work in our lab, and sometimes our mini prep columns basically expire, so we often will pull out the filter of a mini prep column and stuff our cut bones in there. An alternative to this would be just to use a pipette tip and cut the tip of a pipette tip and fill it then with a couple bones, and then drop it into an Eppendorf tube and centrifuge out the marrow. Typically, when we do this, I'll add 200 to 300 microliters of media to the Eppendorf tube and then just centrifuge it out. And it's actually quite surprising how well this works. And then again, you would have to strain resuspend it. Basically, you'll see a nice big red pellet, obviously because most of the cells in there are from the blood cells. But then you resuspend it, and then you'll put it through a cell strainer as well. So this brings up potentially a topic that I think is of interest for coming up with the idea of are bone marrow stromal cells really rare, or is this sort of multipotent progenitor really rare? And the reason why I sort of bring this up is because certainly from the fate mapping studies, you can label lots of stromal cells, and you see a lot of them, and so the questions that you have are they all really multipotent, which we really don't know the answer to. Excuse me. And are they really rare? So I think classically we have always thought of this population as rare, but a lot of the sort of certain pieces of literature have come out that really suggest not only just the fate mapping, but different types of strategies to isolate that really suggest that maybe they're not so rare. Maybe we're just not isolating them in the same fashion, in the right way, or the most effective way. So for example, this is a very interesting paper by Paul Simmons's group where he does what's called a bone marrow plug. So he uses a very small volume of fluid to sort of push out the marrow. And then he carries out a series of enzymatic digests. And when he does this, and the concept behind this is that stromal cells are tightly adherent to basically the bone marrow vasculature. And so the idea behind this is that if you actually carry out an enzymatic digest to try to remove the stromal cells from the actual vasculature, then you'll be able to release more stromal cells. And he shows in this paper how very effective, and in fact, the number of CFUs increased by 2 orders of magnitude, essentially using this kind of approach, okay? There's another work from Paul Simmons' group that was actually done earlier using a different methodology to isolate cells from compact bone. Now this is more, maybe not necessarily stromal cells exclusively, but could be cells also on the bone surface as well. And so they literally use a mortar and pestle type of approach to sort of break up the bone, and then followed by digestion. So the other end of this is to say, well, we flush the bone marrow, but notice that maybe not all the stromal cells are adherent. Maybe there are stromal cells that aren't adherent. And certainly this paper makes the case for that, where they actually take the cell fraction that doesn't adhere, and then they actually go ahead and then they put it on Matrigel, and they're able to find multipotent stromal populations there as well. So I think these are all potentially pieces of evidence that the number of mesenchymal stromal cells may not be as rare as once thought. So when we do basically a culture, I mentioned obviously that lots of cells attach, not only stromal cells, but cells from the hematopoietic lineage. And so I thought I would talk about this because at least when I was a postdoc in training, sort of the jargon was, well, anything that attaches is a stromal cell, which of course is not true. And I think that's because the field often oversells the selective attachment of stromal cells as a way to isolate them. And so in the mouse, certainly, it takes about 0 to 3 days to actually go through attachment when we do culture. Human, it might take shorter, although I'd have to say from talking to people, I don't know if it really is that much shorter. When we culture the stromal cells, we're not using any particularly defined media. We use MEM alpha based media, 10% FBS, and penicillin streptomycin. And what you're looking at here on the right is actually what I believe is perhaps the 2 populations that really inhabit much of the actual stromal culture, at least at first. And the red cells are stromal cells, and so these are labeled with a reporter, an Osterix cherry reporter mouse. And the green cells are actually labeled with this E. coli particle called pH rota. Okay, and you can buy this commercially from a Vitrogen. And this is a very handy and simple way to basically illuminate a lot of the monocyte and macrophage populations in the culture, many of which are adherent. And you could see essentially how there's a ton of green labeled cells, The stromal cells themselves don't uptake the E. coli particles efficiently at all, but the monocytes and the macrophage-like progenitor cells do very effectively. And so you literally just add a little of this bioparticle into the culture under normal culture conditions, and you'll get these cells to literally fluoresce within about 5 minutes. It's very rapid. And so the particle itself isn't fluorescent unless it actually gets taken up into a lytic vesicle, an acid compartment essentially within the cells, and that illuminates the fluorescence. So that's one demonstration showing essentially the very mixed culture of cell populations. But if you look at the flow data here from a day 6 stromal culture, you'll see essentially on the y-axis here on the top panel is a FACS analysis where we stain stromal cells with CD11B, a myeloid progenitor marker, and the x-axis is our Osterix cherry reporter as well. And so you could see essentially the CD11B population here getting pulled up represents 22.4%, and the Osterix population is 49%. And this is at day 6. And I want to say that the stromal population, we'll get into this actually later, but the stromal population early on does not really represent a high number of cells in the culture. The CD45 is another hematopoietic marker, and you could see essentially that the percentage of Osterix cells didn't really change at all, and that the other half of the culture is really composed of hematopoietic cells. Okay, and that's important, I think, from defining the culture, because when you think about isolating stromal cells from different patients, depending on the conditions, the health of that patient, whether they have some sort of disease or condition, the other cells, obviously, in the culture can potentially influence the stromal cells and how they grow and differentiate. So going back to essentially determining the frequency of stromal cells and the colony-forming unit assay. And I will confess, I'm not a big fan of the colony-forming unit assay, and yet it's here to stay. If you try to publish a paper involving stromal cells, 9 times out of 10, a reviewer will demand that you do a colony-forming unit assay. The reason why I'm not a big fan of it is for a couple reasons. For starters, it really just is an assay to a greater extent of growth. The cells in the colony can undergo differentiation while they're in the CFU assay. And there's another problem that I have. Stromal cells really do not like to grow as individual cells. And this is something that I don't think has been pointed out in the literature quite a bit. And that's why I think growing as mesenspheres might actually be a good idea, because once the cells 1, 2, 3 start to multiply, they have each other to attach to. So if you actually look at the methods to grow stromal cells in CFU assays, you'll see some really the necessity to grow them under alternative conditions. They might have to add certain growth factors like PDGF, FGF, or EGF, very common to grow, and it basically comes down to attachment and survivability of the actual stromal cells to form CFUs. One of the ways that we really like to actually culture stromal cells for our CFU assays is in low oxygen. This works actually very well, but interestingly, not only does the oxygen need to be low, but the CO2 level needs to be high. So make note of that 5% oxygen and 12.5% CO2. This makes the media a little bit more acidic. Not clear to me why this is important, but I've done it both ways, where I've done it under normal 6% CO, 5%, 6% CO2 versus 12.5%, and it works a heck of a lot better And in fact, I don't even know if you really see a benefit if it isn't at 12.5% CO2. So obviously, when you plate these CFU assays, you want to plate a very low number of cells in a large area. So the 1 times 10 to the 6 cells is basically in a big 100 millimeter or 10 centimeter dish. The hope is that every CFU is created by a single cell. And plating at low density certainly promotes that, that possibility. I will say that because stromal cells are adherent to vasculature, that sometimes you get bits of stromal vasculature type, sort of like a cobweb of cells type of thing, that won't get strained out, will still come in through your culture and will form a very beautiful colony, but not necessarily be derived from a single cell. So that's just something to be aware of. Okay. So why do I think stromal cells like to basically be in contact with each other? And largely, a lot of what I say is observational, okay? And it has to do with sort of the phases of the culture. The first phase of culture that we were talking about before is attachment. And during this attachment, you have some cell growth from the stromal cells. And in fact, this lower panel here is basically looking at a CTGF reporter mouse. And we published a paper last year showing how this CTGF reporter labels a population of bone marrow stromal cells. And you could see this time lapse essentially over a day from day 2 to day 3, where these 3 cells essentially will divide into 6 cells, okay? So there's some division certainly going on early on, but not a great deal. If you look at the flow diagram, though, going from day 4 to day 7, you'll see that the population increases from 16.8 to 45, almost 45%. So it's really from day 4 to day 7 that you have this really dramatic increase. And when you actually observe the cultures, it's day 4 is when you basically start to see the cells come in contact with each other and start touching each other. So I've always thought, and I think it's generally true, that cell-cell contact between neighboring stromal cells really promotes their proliferation. And in some ways it may make sense. Certainly there are a number of pathways like Adherent and Notch that may drive not only the stemness, but also the proliferation of stromal cells. So how do we differentiate stromal cells? Well, as I mentioned, trilineage potential is essentially into the adipocyte, and the osteoblast and chondrocyte is essentially what you commonly see in the literature. We recently had an investigator visit our university that was able to direct stromal cells into the smooth muscle. And if one of the interesting things about that is that the fate mapping from the Osterix Cree studies did fate map into smooth muscle present in the actual bone marrow. So I think it's very feasible that certain types of muscle, like smooth muscle, make perfect sense. Others have reported cardiac muscle and neuron. I don't know how well this is accepted. I know certainly stromal cells or MSCs from the bone marrow have been used essentially for trying to repair hearts after a heart attack. And I don't know if it's more the sort of anti-inflammatory or some sort of beneficial trophic factors that are being delivered, and not so much the direct ability of the cells to differentiate into a specific type of cardiac muscle. So So you can certainly differentiate stromal cells in their myeloid components or hematopoietic into the osteoblast lineage and adipocyte lineage. But our experience certainly has been for the chondrocyte lineage, to get really good chondrocyte differentiation, you really do need to sort stromal cells. And so this goes back to having a way to sort stromal cells versus passing them. I think the field typically has gone through the concept of, well, we'll purify our stromal cells by passaging them, because as you passage, the number of stromal cells increases and the number of contaminating hematopoietic cells typically decreases. But there is certainly a problem with passaging stromal cells, and that is that they lose their multipotent properties. And this is a work we've done with essentially when we were studying sort of twist-2 Cree labeling of bone marrow stromal cells. And the reviewer asked us basically to carry out sort of a study where we would go through the passaging of stromal cells consecutively and compare the actual multipotency. And you can see there's a considerable drop-off of stromal cells as you passage. And it's not necessarily, you could probably get away, and again, this is mouse, and probably in human you can get away with further passages. And a lot of this may also come down to how you're culturing your stromal cells as well. Anyways. So if you're going to sort stromal cells, you know, what's the best time to sort the stromal cells? And so our experience has been that the viability from the bone marrow, getting really viable stromal cells from the bone marrow has always been a problem. And even from early stage cultures, it's always been a problem. However, we found that starting about day 5 of culture really works very well for us. And we're able to, day 5 is also nice because we can get a fair number of cells to work with as well. So we typically use different reporter lines to sort for the stromal cells, but there are a number of strategies out there that are very effective at identifying stromal cells. There's what's called the negative selection strategy, which largely is using cell surface markers that identify hematopoietic cells, such as CD45, and use that as a sort of subtractive approach and take everything that was not stained as being a stromal cell. And that's a reasonable approach. There's the positive selection strategy, which isn't probably perfect because there aren't cell surface markers out there that exclusively identify stromal cells. CD105 is probably one of my favorite cell surface markers because it doesn't mark any hematopoietic cells and is very selective for the stromal population, but it marks about half of the stromal population, not the whole stromal population. Recently, leptin receptor and PDGFR alpha and beta have also been shown. PDGFR alpha and beta have been out there for a long time. I confess that I've purchased many PDGFR alpha and beta antibodies, and none of them work very well. Yes, they're selective, but it's very hard to get good separation because they just don't illuminate stromal cells. And I don't know if it's a matter of us doing the staining wrong. I think we're doing everything right. Other markers look just fine, or we're just not using the right hybridoma clones. But we've never had very good success with PDGFR alpha because it would be a wonderful cell surface marker as well. There's also other cell surface markers that have been out there as well, like CD51, that we have yet to try. The other strategy, you have negative, you have positive, is a combination strategy. And this strategy works pretty well as well. And so you could combine CD45 and SCA1. SCA1 isn't entirely selective for stromal cells, but if you actually have a SCA positive, CD45 negative population, that marks stromal cells very well. So I want to comment on how to replate stromal cells, because this gets back to the idea of, reinforces my belief that stromal cells like to stay in contact with each other. So if you go and you plate purified stromal populations at low density, where they are clearly all single cells and they're not touching each other, and you grow them under normal ambient oxygen, okay? Not low oxygen or normoxic, but just normal cell incubator, that you will basically have cells that attach fine, and then they won't really grow very well at all, if at all, and the cells will just get larger and larger and larger and form like a big sheet, okay? In contrast, if you take cells and you plate them in what I would call a confluent spot, so you can take a small number of cells, and again, we do this for differentiation, so we take and we plate cells, maybe about 5,000 up to 20,000 cells in a 20 microliter drop, and within the next hour or two, those cells attach and they're in touch with each other, and they maintain their mesenchymal morphology beautifully, and they grow like crazy, and then they differentiate extremely well under those conditions. And so for our assays where we want to basically differentiate into down the osteoblast lineage or the adipogenic lineage, this is what we typically do. We will sort, and then we will literally do what my graduate student says is spot cultures. We will basically plate them in small volumes where basically they're allowed a couple hours to attach, and then we add media onto it after that. And then most times what we do after a sort is we will wash the dish with PBS a couple times, because sorting, when you sort cells, there's always the risk of some sort of contamination from the sorter. And so we tend to wash the cells a couple times with PBS to just limit that possibility. And then essentially culture them as normal. For chondrocyte differentiation, we plate a lot more cells in a smaller volume. So we will plate basically 100,000 cells in a small 10 microliter drop. And the concept behind this is that what you're trying to promote is more of a hypoxic environment. Okay, so it's basically like a little micromass. Okay, so here are the cell media components. I won't go through this in great detail, but essentially you have, you could certainly go back to this webinar and copy down these components. There's no mystery here. These components are in the literature too, in terms of we're not using any kind of magic cocktail here for our differentiation strategies. So as I mentioned before, characterizing differentiation is essentially a combination of whole mount and gene expression analysis, at least in vitro. And so for osteogenesis, we typically like to do the von Costa staining method. It's a very rapid way to essentially detect bone mineral. But even under DIC optics, I hope you notice that you can often see mineral and matrix deposition in your cultures. And when you don't grow them with ascorbic acid and beta-glycerol phosphate, you're just not going to get that kind of matrix and mineral deposition. Okay. When we pick gene markers to assess gene expression, we like to go from beginning of the lineage to late start of lineage. So you can see Osterix is an early gene marker, BSP is an intermediate, whereas osteocalcin and DMP are late markers. So try to pick a variety of markers going from early to late. Often you'll see papers where they really focus on early markers of differentiation and not late markers of differentiation. You want to provide evidence that you have a pretty wide breadth of differentiation going on in your osteogenic assays. And again, note the fold increases of these gene markers. And that's true not only for chondrogenesis, where you basically pick from early to late, but also adipogenesis as well. So anyways, so, and again, for adipogenesis, oil red O staining is a very common approach. We have a variety of gene markers, adiponectin, perilipin, adipsin, and FABP4. Certain gene markers like perilipin are particularly fairly selective for the adipocyte lineage and a really good gene marker to essentially use in your assay. Okay. So I would just like to comment, even though there are cell surface markers out there, that there are really nice strategies now, not only generated or highlighted by some of the work in my lab, but other labs that are capable of labeling stromal cells. So I recently published a paper on connective tissue growth factor, reporter mice. And so right next to these reporter mouse lines, I put also a PubMed ID. So you can use this ID to essentially search, to find papers that use this type of strategy to label Stromal cells. And so there are sort of gene-driven reporters, and then there's the Cree reporter type of strategy that I included in here too. And I would encourage you, if you're going to use the Cree reporter strategy, to use the AI series of Cree reporter mice. So the AI stands for Allen Institute, and these were Cree reporter mice generated within the last 3 or 4 years that really are spectacular. My lab has used a variety of Cree reporter mice, from Rosalac-C to ZEG to MTMG, and none of them really, in my opinion, compare to the AI series. There's AI9, AI14, there's different colors of tomato and green. So if you're going to use this strategy to label stromal cells, I would encourage you to try to use this kind of Cree reporter strain. The benefit of using that strategy, honestly, is particularly for tracing. So you not only label the stromal cells, but out of the bone marrow, but then when you go and do your transplantation studies, you can essentially trace them into other lineages as well. So that's the huge benefit there. So going through the real-time reporters, connective tissue growth factor re-reported hits a stromal population that seems to inhabit the trabecular bone region for some reason. And this is a reporter where works very well in mice that are 6 to 8 months of age. And then the fetal calf serum, when you grow stromal cells in fetal calf serum, will also turn on this reporter as time goes on. Smooth muscle actin is actually another reporter that's very good at labeling stromal cells. This is work done by Ivo Kolagic's lab. And however, smooth muscle actin does not do, in my opinion, a very good job at labeling stromal cells live in the bone marrow. Okay, so when I section through this animal model and look at the bone marrow, you don't really see any stromal cells labeled. You see smooth muscle on the actual blood vessels very nicely, even in the bone marrow, but the stromal cells aren't labeled. However, when you actually culture these cells, over the first 5 days of culture, the stromal cells start to turn on the reporter very nicely. Osterix cherry reporter mice is a reporter line that my lab generated, and it's an interesting mouse model. In tissue section, it does not broadly label stromal cells. However, by the bone surface, we've noticed that there is stromal-like populations that are expressing low levels of Osterix. And when you flush the bone marrow, you do see Osterix positive cells that attach and are expressing the reporter, and then this reporter expression stays at low levels in many stromal populations until you induce osteogenesis. CXCL12 and stem cell factor are known stromal markers, not developed by my lab, but there are animal models that are out there in the literature as well. and are very good at labeling stromal cells as well. Okay, and I would just like to, as just the last note, acknowledge essentially some members in my lab. Yaling Liu is sort of research assistant in my lab that's done a lot of work with stromal cells, and Sarah Stryker is a former graduate student of mine, actually, that has actually done a lot of beautiful work with stromal cells in the past as well. And then, of course, I would like to acknowledge my funding sources, the NIH, internal support from the UConn Health, and even the Connecticut Department of Public Health as well. So thank you. Amanda?
Thanks, Peter. That was an excellent presentation. And so now we have a time for just a few questions. So I guess one of the questions is, how does aging affect stromal cells?
All right, it's a very good question, and there's I haven't worked particularly in this paper, in this area, but there are a number of people that have, and the general consensus is that the problem with aging is obviously stromal cells senesce, and so there's greater senescence, and so what happens when stromal cells senesce is their capacity to proliferate is substantially decreased, and their capacity to differentiate is also substantially decreased.
Okay, that makes sense. And so I guess the other question is, do different bone marrow niches, influence the differentiation of stromal cells?
So I think in general, this is a great question because a lot of the time there's this tendency when we take stromal cells out of the marrow that we don't really understand the variety of niches that exist in the bone marrow. The hematopoietic field certainly talks about niche, but it's always from the standpoint of the hematopoietic stem cell, and not necessarily from the standpoint of the stromal cell. And so I think one of the things that, since I'm interested in skeletal biology, I'm always very interested in the stromal cells that are potentially primed to become osteoblasts. And so if you look actually at the endosteal bone surface, essentially, you have obviously a combination of cortical bone and trabecular bone. And the trabecular bone actually is a very active place of resorption and formation. And so that environment is very different than cortical bone, where you look at the mid-diaphysis and you don't have very much active bone formation at all. And so you can imagine that, there are stromal cells in the trabecular bone area that may be more primed to become osteoblasts, and in other areas of the bone be more primed not to become osteoblasts, and in maybe favor adipogenesis. And in fact, there are some studies that show how, certain locations in the bone marrow where adipocytes are notoriously formed versus others. So for example, if you take a tibia bone and do a tissue section through a tibia, typically the tibia's distal end typically always has much more likelihood of having adipocytes in the bone marrow at its distal part, but not at the proximal end where the trabecular bone is. But eventually, I think as we age, we all succumb to having a lot more bone marrow fat. So yes, there are different niches, and I think there is sort of this, what I would call almost, because of that environment, a predisposition of cells to differentiate along one lineage versus another. And it's not to say that they couldn't, that a cell that was, what I would say, leaning into the adipocyte lineage still may have the capacity to become an osteoblast, but may take longer to do so under the right culture conditions.
Okay, and so we've got a question from Jennifer asking, do you remove red blood cells from the bone marrow aspirate before plating for the CFU assay?
We typically don't.
Okay.
No, we don't. I know certainly some people do, and there's people who use Ficol gradients to remove the red blood cells. Ficol gradients, I don't think, really do a good job at separating, or even sucrose gradients, separating sort of MSCs or stromal cells from essentially other white blood cells. But no, we don't typically go through the efforts. When we do flow, sometimes we will go through an RBC lysis, certainly. Yeah.
Okay. And then Ankush asks, do stromal cells express the lineage marker? So would a combination of, I think it's SCA1 positive and the negative sort stromal cells or hematopoietic stem cells?
I'm trying to see if I understand this question. Can you repeat that again?
Okay, sorry. Do stromal, so his main question is, do stromal cells express the lineage marker?
Well, I guess the question is what lineage markers? So there are a variety of lineage markers out there. So I think often when people talk lineage, they talk about the hematopoietic lineage and say lineage negative versus lineage positive.
Right, that's what he's talking about.
Okay.
So, yeah.
So that's a great question, actually, because CD45 in my book is a little bit, at least when it comes to mouse, I don't know about human, directly from the bone marrow may be a problem, okay? And so there is some work, some evidence out there that CD45 actually may not be necessarily exclusive to the hematopoietic lineage when you try to get stromal cells directly from the bone marrow, and that there might be a small fraction of stromal cells or even early osteoprogenitor cells that express CD45. However, you actually culture the cells for a short period of time, such as 5 days, and typically those lineage markers will rapidly go away. Okay.
Okay, and then Jennifer asks again, do you remove the non-adherent cells from the CFU assay culture?
Yes. But again, the length of the attachment is typically, we still will allow, 0 to 3 days, about 3 days for attachment. When you're plating in CFUs, eventually you got to change the media, of course. And 3 to 4 days later is typically when we change the media.
Okay. Well, I think that's the last of our questions.
Okay.
So that brings us to the end of this webinar. So thank you again, Peter, for a very illuminating presentation and a great discussion. And thanks also to our sponsor, Bio-Techne. And finally, thanks to you, the audience, for taking the time to attend and listen in. If you've enjoyed the seminar and would like to view the video recording of the session, please visit the webinars page on bitesizebio.com. It should be available within the next 24 hours. There, you can also see the other webinars we have lined up for you in Bitesize Bio's Webinar Festival. So until next time, good luck in your research and goodbye from all of us at Bio-Techne and Bitesize Bio.